Hey, everybody, we're going to get started here in just a second. Right. So today we will be reviewing chapters 2 and 4 and going over just a couple of questions from each of those chapters for our most missed topics. I think we've got one for chapter 2 and maybe three for chapter 4. I'm not really sure. So we'll see. So, reviewing chapter 2 stuff, we learned about the different types of bonds. And so, here it is important to remember that what the different types of bonds are. And there's three major ones that we talked about in our chapter, in our course. I know we learn about van der Waals forces and stuff like that in chemistry. And I'm not saying those aren't important, of course they are. But the three that we were most concerned about for our course were covalent, ionic, and hydrogen bonds. Covalent bonds, we are sharing electrons between two different atoms. In ionic, we have an actual transfer or donation of electrons from one atom. There's an N in there. I remember. Transfer. All right. Let's see if it's not going to let me erase it, so I'll just rewrite it. Transferring or donating. And then hydrogen bonds are just attraction forces. No actual exchange of electrons there, but just an attraction between this monitor is so wobbly. I don't know if I can stabilize it any more than it is. It's really annoying me. Um, Attraction force between an H that's bonded to something that has a positive polarity, attracted to something that has a negative thing. We'll put R just to represent some general thing. Okay, it's usually an oxygen that has a negative polarity to it, but it could be anything. And that this itself, that attraction, I forget about this pen not wanting to let me draw things. That attraction there is what's going to be the hydrogen bond. So between hydrogen and some other kind of atom on another molecule, that attraction. But it's not both hydrogens, right? All right, organic molecules. These are things that are carbon-based. That's the key there. And the general other definition of something that is going to be organic, we have to have hydrogen bound to the carbon somewhere. That's a requirement for an organic molecule as well. So it's not enough just to have carbon there. That's why carbon dioxide is not an organic molecule. Carbon dioxide is not a polar molecule either, but there's reasons for why it's not organic either. And we're not going to go into that discussion in our course. That's deep. That's deeper than what you even need to know for chemistry. But that's a physical chemistry thing. And so then that's talking about physics and how those electrons are moving and how they're interacting. But our general idea of what makes an organic molecule organic, it deals with carbon binding with hydrogens. And then that sharing of electrons is very equal and respective to those atoms. And that's what makes something organic. All right, polarity and hydrophobic or philic. Here, when we're talking about polarity, we're usually talking about hogging unequally of electrons, right? And so something that is has a tendency to hog electrons like oxygen does, we call that electronegative. And so it has a negative polarity. Something that is more likely to have electrons hogged from it by things like oxygen than have a positive polarity, that would be something like hydrogen, for example. All right. Hydrophobic and hydrophilic are exactly how they sound like. Hydrophobic or phobia would be like fear, and hydro being water. Now, it's not actual literal fear of the water, right? But it's being repelled. It doesn't mix well with the water. Things that are hydrophobic tend to be non-polar. Things that are hydrophilic, water-loving, tend to be polar. And we mean that in the charge sense, just like we were talking about with our unequal sharing of electrons. So things that are polar have unequal sharing of electrons. And that often means that an oxygen atom is hogging electrons. And now that oxygen is more electronegative. It's got a negative charge to it, negative polarity, and sometimes can have a true actual charge because of ionization associating with that. All right. All right, moving on to pH, which we've covered quite a lot in lecture as well as in lab, but briefly to remind everybody without me having to re-review everything about pH. pH being, and I'm going to rewrite the thing so we can see what we're working with here. pH means the negative log of the concentration of hydrogen ions. That's what's actually pH. And so if a pH is 6, this means that the negative log of the concentration of the hydrogen ions is 6. And what that really means and what's going on there is that the hydrogen ion concentration, in order to get 6 as the pH for the math to work out, that actual concentration of your hydrogen ions is 1 over 1. Oh, come on, work with me. I'm just trying to zero. 1, 2, 3, 4, 5, 6. Or 1, 2, 3, 4, 5, 6. That's the actual concentration of the hydrogen ions for the pH number to be 6. If you plug in 1 over whatever this is, a million. If you plug in 1 over a million as the concentration, the negative log of 1 over a million is 6, because log is a base 10 thing. You can also look at this as 1 over a million is the same thing as 10 to the negative 6. Those are the same thing. These are all the same numbers right here. All right, and that's where we're getting the 6 from. The negative log of 10 to the negative 6 is 6. Okay, so the negative is talking about it being an inverse. The log is talking about it being in base 10. And then we can apply those rules to our concentration of hydrogen ions. And then that gives us the pH. So the more hydrogen ions you have, the lower the pH. More H plus equals lower pH. This is where we get that idea with our acid on our pH scale. Being, if we have our pH scale, we know it's 0 to 14. We know that 7 is neutral. We know that that's the pH of water, right? And that's because water dissociates like equal amounts of H plus and equal amounts of, good job. I have to erase that now. I have to do it. There's no scratching that one out. OH. And then if you have equal amounts, that's why it falls in the middle. Okay, so everything is literally pH. It's based around water. That is literally what pH is based around. And if you get to 0, what that really means, a pH of 0, means the pH equals the negative log of 10 to the 0. And if 10 to the 0, if we're talking about that, is 1, then that means 100% of your solution is H plus. You can't get more H pluses than 100% of your solution being H pluses. All right, the closer you get to that, the more H pluses you would have. And then if you have enough H pluses, like as we're getting near to that pH of 0, the more likely it is that it's going to disrupt your chemical bonds in biological molecules, like proteins or DNA or whatever. Which whenever you see alien movies, which if you guys had lab with me today, you heard me talking about alien movies, but you see the alien movies and most of us know the aliens have the acid for blood and it eats through even the spaceship and whatever. And there are acids that are that strong, like hydrofluoric acid. And that hydrofluoric acid, that fluoride in there, fluoride is one of the very few atoms that is more electronegative than the oxygen. So not only do you have the H pluses, but you also have the fluoride that is super electronegative. But even if you have hydrofluoric acid, would that be a pH of 0? I hate to break it to you guys, but no, because you have fluorines in there too. So it's not possible to have 100% H pluses if you also have other things in there too. So that's another thing, another way to think of that, right? So it's almost impossible to get to a pH of 0. I'm not saying that it's totally impossible. We could create it, forcing it, but it's not naturally going to happen. But as you tend that way, the more likely it is that an acid would eat through skin, eat through pretty much anything, because all those H pluses will like basically interact with all sorts of things that are just holding molecules together and how they interact with the environment around us, which is largely governed by polarity and charges and things like that, right? Okay. Do we have any questions about pH before I move on to the next topics? Because I know that's a one that, I don't want us to get lost on that. Does anybody have any questions about pH?
Professor Yost, this is Brandon. We
Yep.
talk a
Oh,
lot
hold on
about
real quick,
the.
Brandon. All of my sound is coming through my headphones,
Can you hear
and
me?
I don't have my headphones on. So give me
Oh,
just a second. Let me see if I can pull up my sound on my. Hold on. I got to change it to my speakers, and it is not letting me. I'll keep it really quick. Give me a second. I don't want to put my headphones on because I can't talk. It doesn't come through clearly sometimes on that. So just give me a second. All right. Go ahead, Brandon.
um, I just so the the H plus ions are pretty important. Does the OH, the base, do we have to do anything with that?
I don't, pH, I hate to break this to you as well. So is pH affected by the OH molecules? Of course it is. But pH itself, what is it actually measuring? The H
Just the
plus.
H plus.
And so that's all we have to worry about for my class.
Okay.
Now
Okay.
we learned about the OHs and how if you introduce OHs, which are base, right? The
Yes.
OHs basically eat up your H pluses, but as long
Okay.
as you understand the H pluses and what they're there for and what they're doing in there, then you understand the concept of the pH. Does that make sense?
Yes. Okay.
Yeah.
Thank you.
Always keep it in the perspective of the H pluses because that's really where the OHs are even important, if that makes sense.
Okay.
Yeah.
Thank you.
Yes. Good question, though. And luckily, I won't make you do the math of these things. Like I said, I won't make you calculate out pH, but the only reason I'm going over this is to put perspective on the pH scale so that we can understand why, when you go lower in pH, you actually have more hydrogen ions there. We did learn it in chemistry, but it is what it is. Now my slides are like this permanently. That's okay. We'll move on. So our next one is our biological macromolecules and our protein structure. So hold on, let me put in a blank. Okay, so we can do this on a separate screen. All right, biological macromolecules. And then we'll do our protein structure next. All right, so biological macromolecules, we know what the four of groups of them are. And at least this one's pretty straightforward, right? We know we have our carbohydrates. We know we have our lipids. We know we have our proteins, which we'll come back to. And we know we have our nucleic acids. All right. With our carbohydrates, these are our monomers for this. So all of this these are big proteins, right? And each of them have some sort of monomer as their subunit. In carbohydrates, we call this a monosaccharide. Fancy word for sugar, right? When we talk about some of the sugars, we talk about glucose or fructose. Those guys are monomers. Those are one like unit of a ring making up of the sugar, right? And those are both 6 carbon sugars. But when we start talking about things like lactose, for example. Lactose is a combination of glucose and galactose, which is two different sugars linked together. So that's a disaccharide. And so then those words and names start getting fun and very confusing, and they don't help describe at all what they are unless you've really studied into that stuff. And I'm not going to test you on that stuff. But being aware that that suffix os is dealing with the sugars, that'll help and that'll get us a long way. We'll later be talking about some obscure sugars, and knowing os means sugar helps. So when we talk about ribose or deoxyribose, those oses, those are sugars. And both of these are 5-carbon sugars. Obviously, one of the big ones that we'll be talking about when we get into making energy for the cell is glucose, which is a 6-carbon sugar. And we'll talk more about that in chapter 10 when we review that aerobic respiration and everything like that. And we're not there yet. Many of these guys, they are carbon and hydrogen, carbohydrate. Luckily, in carbohydrate, it tells you the molecular makeup of what it's made of. Carbons and hydrogens. Hydrated carbons, okay? But hydrated carbons also come with oxygens. And so this has some polarity depending on how that sugar is oriented and how the sugars are oriented within each other. Generally speaking, sugars tend to be polar. They don't completely dissolve in water, they don't ionize, but they mix well with water because they are polar. All right. All right. Next is our lipids. Yes, these are like fats, like fatty acids. We might be talking about waxes. We might be talking about our steroid-based things like cholesterol and many of our hormones. I remembered the name, by the way, my night class. I remembered the name of, we have cholesterol, just to be clear. Eukaryotes, mammals, I should be more specific, okay? Mammals, like us, in our cell lipid bilayer, if we draw our little lipid bilayer there, you guys know the drill. I'm not going to do the double tails because it just is too much work. But you got the little tails sticking down. We've seen them in lab lately talking about that gram stain, right? So here's our lipid bilayer. And I already told you that our bacteria that don't have walls, they don't have a very strong cell shape. Our mycoplasmas, they don't have a very strong cell shape, which we'll review that in chapter 4 too, because their lipid bilayers don't have anything to support the shape of the cell. But when you look at our cells, we don't have cell walls, but do our cells have a shape? Generally, some of them may, but many of them do have a general shape. And so that's because it's actually supported, and I'm not going to draw it because y'all, cholesterol is wild. It's got multiple rings going on, but we'll put some triangles in here just to simulate that within our lipid bilayer, we have cholesterol stacked in there, and that gives structure and rigidity to our cells so that they can maintain shape as needed as they build up our tissues and things like that. So we have cholesterol, and then fungi actually have something called ergosterol, which I forgot about it the other day, but they do have that. But these are examples of lipids that are found in biological systems. The lipids, as far as we talk about the monomers that make up the lipid, it depends on the lipid that you're talking about. So if we're talking about triglycerides, then fatty acid tails might be your monomer. So it really depends on what lipid you're talking about when we're talking about the monomers. So I don't get specific on what the monomers are for lipids. But the major characteristic of the lipids that we know and love, well, at this point, I'm going to try to draw a star over that messed up line. There we go. The thing that we know and love about them is that they are hydrophobic. And that's water fearing because they have long, well, that was supposed to go to another place, because they have long non-polar hydrocarbon tails. That's the defining thing. So they got a lot of hydrocarbons. It could be tails or it could be rings in the case of some of our waxes and our sterols, but they've got a lot of hydrocarbon action going on, a lot of equal sharing. They are mostly non-polar, and this usually lends to them not mixing well with water. Polar things mix well with water. Non-polar things do not mix well with water. So much so that no matter how much you shake that bottle of oil and water, that's always going to separate ultimately. So. All right, moving on. Talk about our proteins, okay? Proteins, we know our monomer is going to be amino acids. Come on, thing. And amino acids generally look like this. We have an NH2 group. I drew it sideways so that we could draw our link to our carbon. There's our NH2 group, which is our amino. We have a central carbon. It's linked to a hydrogen. Remember, carbon always makes 4 bonds. That carbon, that central carbon, is attached to some sort of group that defines what amino acid you are, and we call that the R group. And it's also attached to a carboxyl group. You guys remember carboxyl. We got a C double bonded to an O, as well as single bonded to an OH group. That H can come off and leave that O out, which is why this is the acid part of the amino acid, because we have carboxylic acid here. All right, so that's why it's an amino acid. And how we build these guys is that this O that can donate off that H can bind to the next NH2 group. It's to the N, but we're going to move on from this, okay? But it can bond to that next NH2 group. And then another amino acid is there with that R group and that carboxylic acid there. Okay, so that's that link right there. To remind you guys, is called a peptide bond. A peptide, in colloquial terms, when we talk about building proteins and linking amino acids together, peptides are like small chains of amino acids linked together, and which we see in peptidoglycan, right? Creating the bridge between the glycans, that string of sugars, and then the bridge of the peptide bond there. So that's proteins. They're just made of linked together amino acids. You don't need to memorize all of the amino acids. And you'll learn more about how the proteins are made and how we get from DNA code of ATCG, ATCG, whatever, and then getting the amino acids from the genetic code. We will learn about that. And I know you've learned some about it, but we'll review that stuff in chapter 6, which is the first chapter in unit 2. We're not there yet, so don't stress over remembering that stuff just yet, okay? There's our proteins. Proteins can come, and we have 4 different levels of structure of the proteins. That first level, which we call the primary level, come on, I'm drawing it. You guys are seeing it. The primary level of protein structure, all right, primary, just so there's no confusion, it's just the amino acid sequence. Just like with DNA, that's like AACTACTG, whatever it is. In proteins, we're talking about isoleucine, methionine, cysteine, serine, glutamate, aspartate, whatever amino acid you have there. Spells out your protein. That's the primary sequence. The secondary level of protein structure is our alpha helices. I'm just going to put HEL for helices. Helix. Helices is the multiple. And beta sheets. They're beta pleated sheets. Now think of these as being, I'm writing too slow for my pen apparently. Think of these as being effects of the resulting of the R groups next to each other. The R neighbors cause alpha helices. If your R group is causing things to twist around, that might cause a alpha helix. But if the R groups are happy next to each other, we might have straightaways forming the beta sheets. Okay, that's what the beta sheets are. All right. And if you were in my class, then you heard me describe this as our primary is basically building your actual ribbon. The secondary is curling the ends of your ribbon or wherever you want there to be a curl in it. So you have areas of curl and areas of straightaway. The tertiary, the third level, this is going to be how the whole polypeptide and all the groups in it are interacting. So things from far across the way might be more attracted to each other than the things immediately next to them, causing them to come together, or might be repelling each other, causing them to push apart. And then things on the inside, because they're all curled up or straight away, might be bent towards each other or away from each other. We can also create what those disulfide bonds. So I'm going to just put S for di-S. This is where we have cysteines and cysteines coming near each other and then covalently bonding or sharing their actual electrons with each other across that protein structure. So I think of this as tying the ribbon. Okay, so we'll tie our little ribbon here. This is where we got curls in the ribbon. This is still the same one ribbon. Some things immediately next to each other cause that, but now we've actually got, this is impossible to do with this pen. I just got to do it quick. Here we had straightaways. Oh my God, it's impossible with this pen. And curls. And now we've tied that ribbon. That's the tertiary structure. The quaternary is taking multiple ribbons or multiple polypeptides, however you want to look at it, okay? But it's like taking a one bow that I'm never gonna be able to draw it, but it's taking multiple bows and putting them on your package. My pen is not having it. So there's that. All right. Moving on. And a lot of this primary, secondary, tertiary, quaternary. The secondary, tertiary, and even the quaternary depend pretty heavily on the R groups, whatever they are, how they interact with themselves or their environment, whether they're hydrophobic or hydrophilic or even fully charged, ionized. You know, we have aspartate and glutamate, they actually are charged. They're not just polar. And so things like that enable them to interact with other groups as well as other polypeptides and their target molecules, whatever they might interact with. It's all determined by those R groups and how they're interacting with their environment. All right, moving on to nucleic acids, we know DNA, we know RNA, and probably one of the other important ones that you're not including in this group is going to be ATP. DNA is all of these, by the way, are monomer is going to be nucleotides. All right. Yes, this is ATCG when we're talking about them, but it's not as simple as ATCG. All nucleotides are a 5-carbon sugar, all of them. I shouldn't be allowed to draw sugars in case you haven't figured that out in lab. We have a 5-carbon sugar that is based, all of them are based on ribose. Now, if you're deoxyribose, you just removed an oxygen. That's all that means. Literally. Okay. So they're based around ribose. If you're RNA, you have full-on ribose. If you have DNA, you removed one of the oxygens. Okay, that's the difference. And that sugar is bound to a phosphate, which is a PO4, which I'm not even going to try to draw, but you guys know already that this is going to be heavy negative charge on it. Okay. And that sugar is also bound to a nitrogenous base. We'll put NB here. And it's fancy. It's got a lot going on. It's kind of ring based. It might have some oxygens in there, and it might have some hydrogens that are bound to oxygens that might have a positive charge. This nitrogenous base is either adenine, which was A, thymine. We'll get back to thymine. Let me go on. Adenine, we'll go with guanine first. Guanine, which is G, obviously. Cytosine. And then either, if you're DNA, you'll have thymine, which is T. If you are RNA, you'll have uracil, which is U. And A is attracted to T or U, and C is attracted to G. That's the basics for now. Chapter 6 will come in at you with the replication, with the transcription, with the translation. Okay. For now, this is enough for our DNA. For chapter 2, for our review on stuff, this is more than enough for that DNA stuff. But the idea here, these where A binds with T and G binds with C, these are just hydrogen bonds. Let me put this into perspective for you, which I did in my in-person classes, but if you're watching this with my online classes, let me put this into perspective for you. DNA, right? Double-stranded DNA helix. I know it looks like a P, but just it's a D. All right. When you take that apart, it's this, right? A bound with T and C bound with G. And that backbone is just repeating sugar phosphate, sugar phosphate, sugar phosphate. On each side, sugar phosphate, sugar phosphate, sugar phosphate. In the middle, each sugar is made to an A. The other side would be a T and whatever. C to G, G to C, whatever, all the way down. But in between each of these, this right here, between that A and the T, that is a hydrogen bond, which put H bond. All of your DNA is just held together by attractions because of hydrogens having their electrons hogged by the atom they're attached to. And then that causing attractions to the next molecule. All of your DNA is held together by hydrogen bond attractions. So when you have too many H pluses building up in the body, H pluses, I know we answered this, some of you guys that have already worked on your chapter 2 activity, this chapter 2 activity asks about this and we worked through it in class. But for those of you who may not have watched that or weren't there or weren't paying attention, you get too many H pluses. Now your G doesn't want to bind with your C. The H pluses are interfering with that. That's your DNA. But this stuff up here all has to deal with hydrogen bonds too. Now your proteins misfold, we call that denaturing, or your DNA misfolds, we call that denaturing, because we can't have hydrogen bonds holding our major macromolecules together. And that's why the flesh falls off of your bone whenever you get acid on you. Pretty crazy stuff that I am here talking to you right now because of these things. And because my pH is where it's meant to be, I don't have too many H pluses. So do not underestimate the significance of hydrogen bonding, pH, and attractive forces, because that's what governs pretty much everything that's going on, including how our stains work in lab, right? All right. Any questions about this stuff before I move on to the next one, which is chapter 4? All right, chapter 4 is our bacteria and archaea. Mostly bacteria. We're just being honest with ourselves here, right? Hey, the key take-home message, these are our eukaryotes, which is the same thing as a prokaryote. All of our eukaryotes or prokaryotes, all of them are either bacteria or archaea. That's it. There's no mammal, there's no helminth, there's no protozoa, there's no fungi, no yeasts that fall into prokaryote or eukaryote. There's no bacteria or archaea that fall into eukaryote. They're very distinct from each other. All right. In fact, our domains are set up that way. We have 3 domains, which we learned about in chapter 1. But our 3 domains are bacteria, archaea, and eukaryotes. They're on their own. Then we have bacteria and archaea really are that different from one another. They're not even at all similar. They just happen to be unicellular eukaryotes, and that's the only reason why we've lumped them together here. The only real thing that we're going to care about with the archaea, I mean, they're just so wildly different. That's a thing to remember. So many features of the archaea are so different from the bacteria. They have different molecules in their cell walls and their membranes, and their DNA is different, and all sorts of their ribosomes are different. They've got a lot of different stuff than the bacteria or the eukaryotes. They're different, they're in their own domain for a reason. And they live in extreme environments. We call them extreme ophiles. We are talking above boiling temperatures, below freezing temperatures, pure salt. They live in those environments, okay? Typically. All right, so that's about all you need to know about the archaea. Everything else really focuses on the bacteria, and that's what most of what we'll be focusing on in the course will be. All right, we talked about bacterial structures, shapes, and arrangements. Remember our two main shapes were cocci, which were the round ones. At least that's easy to remember because cocci has an O in it, that's round. C's are also round. All right, and then we have the bacilli, and guess what? This might have a C in it, but it has I's and L's, which are upright like rods. Cocci are round, bacilli are rods. Coccus is singular, bacillus is singular. All right. Our arrangements, and you see how you notice how I only talked about cocci and bacilli? I won't test you on the other ones. All right. The arrangements that we need to know. The two biggies, and there were more than one, and there were more than one we need to know, but our two biggies were Staphylo, and these are the ones that everybody gets tripped on. Staphylo is bunches. If you looked in my microscope where I had my staphylococci in it, you could very clearly see how they were bunched up. So staphylo are bunches, and strepto, we have a lot of people getting confused with the strepto. They're chains. If you saw this in your microscope, and it generally had a chain-like shape, that's not strepto. That's staphylo. Strepto is only one-on-one cell, like that. One chain. There's no building on chains on top of each other. They look like that, and that is it. Very pure, like this. That's strepto. Sometimes we'll have diplo. Okay, that falls in the strepto category. All right. All right. Then we have palisades, which were our fence posts, like this. We didn't see any much of that. We might have had one or two people that had maybe a palisade or two, but we didn't see a ton of that in our lab. Lab, we mostly saw staphylo. We might have had one or two strepto or a diplo here or there. And we had a ton of no arrangement, and that's a possibility too. Okay. If you are motile and you're able to move around on your media or in your media, then we call that motility. Bacteria only move by flagella. It's the only thing bacteria use for motility. There are 4 types of motility arrangements. I should just call it flagella or flagellar arrangements. Monotrichous, it's one word, but I'm going to just put trichous here so that we can just go on with the prefixes. Okay, monotrichous, peritrichous, amphitrichous. You guys notice how I'm not going off of notes here. I'm just going off of memory, so bear with me. Amphitrichous, we did the ends, we did the over, we did the one, and lophotrichous. Monotrichous looks like this. I forgot about the drawing. I'm just not going to let me do the thing. I do it fast. Okay. Monotrichous looks like this. Peritrichous is covered in them all over the place. Okay. Salmonella does this. Amphitrichous, you got one at each end. These are your flagella here. And lophotrichous has a tuft, a set at one end. Okay. These flagella move in one direction or another direction. It's like a motor that spins the flagella. The flagellas don't move like a whip. They're either spun one direction or the other direction. And when they switch between those spinning directions, that leads to a tumble. You spin your thing one direction and you can run. You switch the direction, it causes you to tumble. And that's how the bacteria control how they move. They will move in response to things in their environment. We call this chemotaxis. Some of the bacteria move in response to light, especially our photosynthetic bacteria, like our cyanobacteria. They'll move in response to light, to the light typically. And we call that phototaxis. So same idea. So taxi, moving, photo or chemo in response to light or to chemicals. Like we talked about in class, like moving to the smell of a cookie or moving away from the smell of poop. Okay, it's the same idea. So they respond to chemicals in their environment and then they run or tumble to get them closer or away from whatever that signal is, right? All right, our cell envelope, we talked about this in lab, so I don't think I need to go over this a ton, but briefly to remind you guys about the cell envelope. If you are gram positive, you have a thick old fat peptidoglycan wall. Your whole cell this is your cell. Well, this is not a very good picture now that I'm thinking about this, but whatever. That cell wall is surrounding a cell membrane. This is a lipid bilayer, which I'm not going to draw again. We drew it in lab. And if you haven't been to lab yet, my night class, then you'll see it here in just a little bit. But this will be your inner membrane, also known as the cytoplasmic membrane, because on the inside, this will be the outside of your cell. I'm just dividing it from everything else above it. Okay. Outside of your cell and inside of your cell. This is your peptidoglycan wall. Right. And the wall itself is a big scaffolding. You have long chains of sugars stacked on each other, and they're linked by these polypeptide chains, these cross-linked chains of polypeptides. So they're just amino acids linked together, making these cross bridges, holding those sugar chains together. And that is how peptidoglycan exists. So we have chains of sugar. That's the glycan part. Stop it, pen. And then these are our peptide bridges, holding that scaffolding together. And this is peptidoglycan. Big old scaffolding, okay? Anyways, so gram positives have a thick, thick wall surrounding their inner cytoplasmic membrane. That inside, that goo is just the cytoplasm. It's the goo of the cell. Separating it from the outside is that cytoplasmic membrane, also known as the inner membrane. And we have that wall around it giving structure to the cell. Now, gram positives, they just have a thick daddy wall, okay? Super thick wall. The gram negatives, gram negatives have a thin wall on their inner membrane. All right. And then an outer membrane. Because that thin wall is just not enough to give them much structure. But so that outer membrane is there to give added structure. All right. Gram positives, they have the teichoic and the lipoteichoic acid adding in to help with this big daddy wall gets so thick it's hard for the cells to manage. So teichoic and lipoteichoic acids are there to manage that. We talked about that in lab, so I'm not going to draw that out here again, but we talked about it previously in lab. But then our gram negatives have that thin, maybe 1 or 2 layers of sugar. They don't need teichoic acid, but because they don't have a ton of stability and they need a little bit more regulation on how things get in and out of the cell and how the cell interacts with its environment, they have these little outside markers that we call LPS, which we learned is also called endotoxin. It's the same. Lipo being fatty, polysaccharide being many sugar, right? So it's a molecule like that. And it's endotoxin. It causes the body to have an inflammatory immune response just because it's there. So So if it's not associated with a bacterial cell and it's been broken off because our body attacked it or something like that, then that's a sign for our body to have an immune response. So that's why we call it endotoxin, because it leads to toxins lead to damage in our tissues. Endo because it's inside, it's part of the gram-negative cell. All right. Other things that make different walls. So you're either gram-positive or you're gram-negative. These are the two that are number one, the most significant aspects. All right. Mycolic acid is something that is a feature of gram-positives, that some gram-positives only Mycobacterium have. It's a special wax, right? It's a lipid that makes these cells more resistant. And it's just a wax that's on the outside of that already thick wall. And that's what it is. Okay. Mycobacterium, it'll be in their name. Mycobacterium, the whole word. This is Mycobacterium tuberculosis. That's the name of that organism that causes tuberculosis. Mycobacterium leprae, that causes leprosy. Mycobacterium avium, which is also another cause of tuberculosis, specifically associated with HIV that's uncontrolled and leading to AIDS. And then Mycobacterium smegmatis, Mycobacterium flea, but they're all named Mycobacterium something. All right. The ones that don't have a cell wall, these ones are type of gram-negative. They're like our gram-negatives here, but they just don't have any wall at all, just the two membranes. Our gram-negative ones that don't have a wall are called mycoplasmas. Focus on the plasma, like I said in class. Plasma, like a cup of plasma, if you spill it, it doesn't hold its shape. Neither do mycoplasmas because they can't hold their shape because they don't have a wall to help them hold their shape. Okay. So these guys are pleomorphic. They have many shapes. They are not staphylo, they are not cocci, they are not bacilli, they are not vibrio, they are not spirochetes. They are really pleomorphic, many shape. All right. So those are our two weird outliers with the walls. All right. All right, moving on to the gram stain. We're talking about this in lab. I'm not going over it again. Just the lab covered it, and I felt like it covered it better than the lecture did. And so remember how the gram stain works and why it works. Remember that the gram stain works because the stain gets trapped in that scaffolding of our peptidoglycan wall in our gram positives, gets cross-linked by our iodine. Alcohol will rinse out anything that didn't get stuck, and that's why the gram positives stay purple. We counterstain with safranin so we can see that our gram negatives are there. And so our gram negatives are red and our gram positives are purple. So I'll put that just to remind you. Gram positive, purple. And gram negative, red. Next is our endospores. Endospores are protective dormant structures. They are completely dormant. They are completely inert. If you are in endospore form, you aren't doing anything. You're not making toxins. You're not multiplying. You're not doing anything. You are literally just sitting there. The only thing that endospores can do is become vegetative. That's it. Vegetative are active. They are multiplying. They are making toxins and infection. The only thing that endospores can do is become vegetative. Vegetative cells are active, multiplying, they're doing metabolism, they're making energy, they're making toxin, they're multiplying, they're growing, they're doing things. The only thing that endospores are doing is nothing. They're just waiting to become vegetative. So when you get sick because of an endospore, the endospore got into your body. Sure, it was hardy, it was resistant, it resisted the cleaners, or it resisted drying out, or it resisted whatever the harsh environment was that made it become an endospore. Then it got into your body, which was nice and warm and moist. And then it was able to turn into the vegetative form and then cause the infection and the disease, right? So it's never the endospore that causes the disease. It's always the vegetative that causes the disease. The endospore got in there, but you don't get sick from endospores. You get sick from the vegetative version. All right. Only two genera can make endospores, Bacillus and Clostridium. Yeah, you need to know it. If I start talking to you about how a patient got sick from eating food that was left out in a heat lamp, and we're suspecting that it was caused by Bacillus cereus, well, even though that food got left under a heat lamp that was warm and other bacteria might have died, the Bacillus cereus made endospores that didn't die from drying out or the heat under that heat lamp, right? And then the Bacillus cereus got into the body and then made toxins that led to the food poisoning, for example. All right. Obviously, we talked about several Clostridium-based diseases like botulism, tetanus, gas gangrene, and C. diff, which is pseudomembranous colitis. We talked about several diseases associated with Clostridium. Clostridiums are also anaerobic; they don't live in oxygen. Bacillus species do; they like oxygen typically. All righty, let's move on. Um, we could talk about the endospore process a little bit. So when you're switching from vegetative to endospore, you're going from vegetative to endospore. This is called sporulation. It's a process called sporulation. One vegetative cell becomes one endospore. It's not going to let me draw it, but you know, you get the idea. All right. And then one endospore becomes one vegetative cell through that process of germination. All right, this is in chapter 4, but we'll also review it when we do the endospore stain, which we don't do before the Unit 1 exam. So be sure you brush up on this before the Unit 1 exam. All right. All right, one thing that I mentioned to my class this morning, especially if you're my online people, if you're just reading my slides, you're missing a lot. Don't be that person. Don't be that person. Because if you've watched my lectures, you will very quickly realize I'm not a slide reader. Have you seen these slides and how much more information I put in my lecture versus what was on the slide? If you just read my slides, it's like just reading these slides. I'm not a slide reader. You're missing a lot if you don't watch the lectures. Just a
Thank
heads up.
goodness for that.
Right? So I that's the case, right? So that's how we feel about professors in general. If they're just slide readers, what are they even doing? Why are they even teaching a class?
Exactly.
You can read slides on your own. So yes, so if you haven't watched the lectures, but you think you can get by with just reading the slides, I'm not a slide reader. So information is on the slides. Of course, that is helpful and it helps you keep track of what I'm talking about. That's what the slides are for, right? And helps me keep on track too.
Sure,
So
yes.
yes, what's up?
So you officially ruined buffet dinners for me.
I gotta tell you, man, and I'm a stickler for it too. Like when I go out to like family get-togethers or whatever, and the food is sitting out, they say 2 hours, right? Because that's all it takes. 2 hours of sitting out at room temperature, and you're in the danger zone. And that's the thing when we start talking about some of these diseases, which will be in it's in Unit 4. We'll get into those types of diseases, but are like there's a difference between having salmonella, which is an actual infection with that bacteria, and then what we call food intoxication, or when we say it's like what do we say? People say they got like a little 24-hour flu bug, a food, gut bug or whatever. Um, and that type of intoxication is from the toxin that those bacteria produce. And here's the thing, guys. All it takes is 1 to 2 hours for Staphylococcus, which is on a lot of people's hands, right? To multiply enough to make enough toxin that'll stay on your food. And they are heat stable toxins. So even if you reheat the food to a safe temperature, the toxin will still be there. Even if you freeze it and reheat it, the toxin will still be there. So if it's been out for 2 hours or longer, don't eat it anymore. Unless you want to be sick for 24 hours. And it's just 24 hours. You'll get over it. You'll get past it. But we've all had it, right? We've all had it. So that's why. So be mindful. I'm picky about it. I will go to potlucks or whatever, and I'll eat when I'm first there. But if that stuff has been sitting out for 2 hours and people be like, oh, take it home. And I'm like, I'm going to take it home and throw it in my trash can. That's what I'm going to do. So I'll even take it home just to throw it away so other people won't eat it. So keep in mind, it changes your perception of things when we start learning about this stuff.
Yes, it does.
All right, so our last thing are our little terms for our 4 different groups of our eukaryotes based on their walls or envelopes or however you want to look at it. So I'm trying to keep on track because I have to leave here at 6:20, but we've got a minute. So we will, let's see, our 4 groups. There's 4 types based on the cell envelope. The first one, let's do it, Mendoza cutes. If you've watched my video, then you know I'm bringing Mendoza cutes up first, okay? Mendoza cutes. Then we have the firmicutes, the gracilicutes. No, I'm throwing myself off because I'm trying to write gracilicute and my pen does not want me to. And then the last one is tenericutes. All right. When I was in middle school, which was a long time ago, okay, this is an archaic story, right? Long time ago in middle school, I had a boyfriend. His name was Joey Mendoza. All right, so that's how I remember Mendoza cutes are the archaea. It's silly, but that's how I remember it. I think of Joey Mendoza. All right, the firmicutes. They're firm because they have the thick cell wall. These are our gram positives. Where would our gram negatives fall then? Into the other G. That's really how I remember it. It's okay. And then that leaves the tenericutes. The tenericutes, tener does mean like soft, like it's like pliable. I mean, basically tender, and tener looks like tender to me. So tener acutes, these are the tender ones. These are the ones without the walls, no wall. And that's how I remember how those are grouped. So when I start telling you that a patient is presenting with anthrax in your hospital and they seem to have endospores, if I say, oh, this patient has a disease, they found endospores. All right, which category would it fall into? If they have endospores, we learned that the endospore guys are Bacillus or Clostridium. As it turns out, both of these happen to be gram positives. And these are the only bacteria that can make endospores, and they're all gram positives. Not all gram positives make endospores, but if they make endospores, they must be gram positives. Okay. Therefore, if your patient has anthrax, that pathogen makes endospores. It falls into firmicutes because those are gram positives. We tracking? I hope so. We're moving on to our examples. Chapter 2, I think this is the only one we have for chapter 2. This is the chemical bonds. Covalent, we are sharing. Ionic, we have donation. And then hydrogen bonds are attractions. These slides have a little bit more explanation on these topics if you need a little bit more explanation on these topics, but I'm not going to read them to you. You are welcome to read through them. And then I sometimes have little mnemonics or ways of remembering things. If you don't like them, don't use them. All right, so I'm going to write our little cheat notes out to the side. We know covalent, we are sharing. Ionic, we are transferring or donating an electron. And we had hydrogen bonds. I want to put bonds, not just hydrogens, hydrogen bonds, right? That these are just attractions between an H that has a positive polarity to something that is negative polarity. That is the hydrogen bond. All right. All right. Let's see if we can answer our questions with that. Bond between magnesium and sulfate in magnesium sulfate allows it to completely dissociate into Mg plus. It's plus because it lost an electron. Now it has less electrons than it has protons. That's why it's plus. And SO4 minus. It's minus because it has extra electrons. I didn't balance these, okay? They're 2 plus and 2 minus, but we can live, right? When we dissolve it in water. You're welcome. What kind of bond exists between magnesium and sulfate? If we have this full transfer of electrons going on, those are ionic bonds. Remember, covalent, we are sharing. Hydrogen, we have attraction involving a H that has polarity of positive. Van der Waals are also attractive, but no H's. And we're not going to test you on them in my course. They will never be the answer to the situation. All right, moving on. DNA exists as a double helix, and it's called double because we have that 2 strands of alternating sugar, phosphate, just like we drew earlier, sugar, phosphate, sugar, phosphate, sugar, phosphate. And we have an A, and it's attracted to a T, or a C that's attracted to a G, right? And this here, we already know because I'm just reviewing. I haven't even gotten to the question yet, but I'm drawing out my thing because this is what I remember. And this is how I take tests. I'll like draw out what I remember and then apply it to that situation. All right. So we call it double helix because we have the 2 strands held together exclusively by hydrogen bonds. In any of those bonds, what type of electron interaction is occurring? Well, we already said these are just attractions. We don't have actual electron exchange of any kind. We're not sharing electrons. We're not donating electrons or anything. So we don't have electron sharing. We don't have electron donations. We have polar attractions, no electron transfers. Last one says negative electrons becoming positive. That's a quantum physics thing that I don't even want to know. We're going to say no on that. All right, into chapter 4 stuff. Understanding the difference between gram positive and gram negative. All right, we have our basic differences here that's just spelled out a little further than our previous slide. I mean, I wrote it out and drew it out, but here it is typed out, so clearer than my writing. But gram positives, thick wall supported by lipoteichoic and teichoic acids. Gram negatives, thin wall, doesn't really support the cell, so it needs an outer membrane. That outer membrane has lipopolysaccharide, which is basically endotoxin. All right. We'll do that. We'll do gram positive, thick peptidoglycan wall. Gram negative, thin peptidoglycan wall. And outer membrane. That outer membrane isn't as simple as just saying it's an outer membrane. It also has LPS, which is the same thing as endotoxin. I know we just did it in lab, but when it comes up in the test, I don't want you guys to forget it either. So both of these have a cytoplasmic membrane. All cells have a cytoplasmic membrane, also known as an inner membrane or a cell membrane or a plasma membrane. That's all the same. All right, let's see if we can answer a question with this. What patient sample is tested? And you see it has lipoteichoic. I didn't elaborate on that, but it was on our previous slide. We elaborated on that. It's not in my cheat notes, right? Lipoteichoic and teichoic acid. They are supporters of the thick peptidoglycan wall. I'll just put teichoics. If it's got some kind of teichoic, then that's the supporting molecules that we find in the gram positives. Gram negatives don't have lipoteichoic. They have LPS, lipopolysaccharide. Acid fast. Now, acid fast bacteria that have the mycolic acid wax on them, the mycobacteriums, they do have lipoteichoic and teichoic acid. But just because you have lipoteichoic and teichoic acid, does that mean you are acid fast? No. That means you're gram positive. And acid fasts are a type, a subset of gram positives that have this extra wax on them. We can tell. Which of the following is not a feature of the gram-positive outer membrane? Trick question. Did I write about an outer membrane for our gram-positives? No, because they don't have one. Their wall has this lipotechoic acid, not their outer membrane. They don't got an outer membrane. Their wall has peptidoglycan. They don't have an outer membrane in the gram-positives. The gram-negatives have lipopolysaccharide in their outer membrane. Next topic is dealing with our endospores. All right. Endospores, dormant and inert. They're not doing anything. They have a thick, what we call a cuticle or cortex, just a thicker layer than just even peptidoglycan. It's other things in there that make them like really resistant to a lot of things and very difficult to get rid of. So they're dormant and they are resistant and they're completely inert, but they're very, very heat stable and all sorts of stuff. All right, that making endospore is called sporulation. Going from endospore to vegetative, which is the reproductive version, is germination. And only Clostridium and Bacillus can do it. Let's draw our little cheat sheet. We know that vegetative is our active form. We know the endospore, often called just spores when we're talking about bacteria. The endospore is inert or dormant, very resistant, hardy. And we go from vegetative to endospore through a process of sporulation, we'll just put sporulate. And then going from endospore to vegetative is germinate. Like a plant will grow. If you start growing from going from seed to plant, you're growing, germinate. Okay. All right, so this is our cheat sheet. A researcher discovers bacterial strain. Oh, and the other thing we didn't put in here, things that can do this, just before we go too far. Bacillus species and Clostridium. I might not make you type out Clostridium, but you should be able to recognize it in a multiple choice. All right. Researcher discovers a bacterial strain that forms endospores. We already know a couple things. It forms endospores. It's got to be one of these. It's got to be Bacillus or Clostridium. It lacks certain genes associated with the coat synthesis. So this is what makes it so dormant and hardy, right? After exposure to extreme heat, this strain has reduced survival compared to normal strains. So now our endospore is not as hardy as it should be. It's dying from that heat, all right? Which of the following conclusions can be drawn from this observation? Is the bacterial's reduced survival rate a result of DNA replication? We're not affecting the DNA, we're affecting how hardy that endospore is because of the coat that protects it. So it's not that. Is it the absence of those spore coat genes impacting the ability of them to withstand extreme environmental conditions like heat? It's probably B. Is it because of reduced survival rate of the spores because of bacterial strains, higher ability, metabolic activity? No, if you have a higher metabolic activity, that doesn't affect your ability to resist heat. Might multiply faster, but it won't help you resist the heat. Strains endospores are likely to be more resistant to chemical disinfectants. It's actually going to be less resistant. If their coat makes them more susceptible to heat, they're actually probably more susceptible to the chemicals too, I'd imagine, but we can't infer that directly. But this is saying the opposite of that. So our best answer here is B. All right. Next question. Patient of yours is diagnosed with C. diff, Clostridium difficile. Why would containment protocol be more strict for C. diff than for Salmonella? Well, first things first, is Salmonella called Bacillus or Clostridium? No. So does Salmonella make endospores? No. Salmonella doesn't make endospores. Well, A is out, right. Clostridium makes endospores. Clostridium difficile is endospore forming, which enables it to be more resistant to cleaning and cleaning agents and stuff like that. So we're going to have a more strict protocol in place to control the spread of those endospores. Do all gram-negative bacteria form endospores? No. Specific gram-positives do, Bacillus and Clostridium. All diarrhea-causing bacteria are endospore forming. In fact, most diarrhea-forming bacteria don't make endospores because most diarrhea-causing bacteria actually are gram-negative. Most of them are. Salmonella, E. coli, Shigella, most of those guys are gram-negative and they don't make endospores. Okay, so B. Next question. I believe this is our last topic for this. The cell envelope. This was the Mendoza-cute, firmicute, that stuff. Okay, so here this is kind of breaking down the differences in our cell envelopes, including our mycobacterium that have mycolic acid, wax, and mycoplasmas that lack a wall. So let's make some notes about this stuff over here on that side. Well, let's just break down the question and I'll give you the notes as we go. A microbiologist is studying a bacterial strain that has a thick peptidoglycan layer in its wall. Sounds like a gram-positive, right? Based on this description, which of the following groups does this strain most likely belong to? Well, it's a gram-positive, but that's not one of our choices. So now we need to understand our choices. The firmicutes, these have a thick wall. They're firm because they have a thick wall. These are our gram-positives. They're firm because of that thick wall. Gracilicutes, I'm focusing on that G. These are the other G, the gram-negatives. All right. The Mendoza-cutes, like Joey Mendoza from a long time ago, these just describe the archaea. Remember, they have weird molecules in their membranes and their walls. And I'm not testing you over what those molecules are or anything like that, but they have the weird molecules, all right? And then the Tenericutes are tender. Okay, tender. I shouldn't be allowed. Tender, because they have no wall. This is specifically our mycoplasmas. Remember the plasma, because we spill it and it doesn't hold its shape. It doesn't hold its shape because it doesn't have a wall. Remember, our gram-negatives have a thin wall and outer membrane with LPS or endotoxin. Just to remind you. All right, so there's our little cheat sheet for our next question. So question 1, thick peptidoglycan layer. Our answer was A. Our second question, 56-year-old patient presents to the hospital with GI symptoms, including nausea, vomiting, and abdominal pain. Laboratory tests reveal that the causative agent is a gram-positive, rod-shaped bacterium. Well, the shape. So gram-positive, if it's gram-positive, it's probably going to go in the firmicutes, right? It's rod-shaped, that shape. Just to remind you guys, we call rod-shaped a bacillus. Now, not all rod-shaped bacteria are named bacillus. Those are different there, all right? For example, Salmonella is a rod-shaped bacteria, but we call it rod-shaped bacillus. If it's bacillus in its name, then it is endospore forming. Okay, moving on. Upon further testing, it is noticed that the bacteria's wall is thick and it shows resistant to desiccation in high temperatures. All right, we suspect the infection might be due to a common bacterium in the soil that can form endospores. So now we think it's an endospore forming thing. This is probably a literally named bacillus something or Clostridium something. It's capable of surviving extreme conditions. This is probably most commonly B. cereus. If we were talking about this is Bacillus cereus, cereus, commonly associated with food poisoning. That's the word I couldn't think of earlier. Food poisoning, 24-hour bug. Okay, goes away because that poison, the toxin, is very limited. It's self-limiting. It doesn't propagate itself. All right. Endospore bacteria. So even though we've already called it gram-positive, if I told you that it had endospores, we should know, okay, endospores. That's either caused by Bacillus or Clostridium, and both of those are gram-positive, which I talk about multiple times in the lecture, and I talk about it in the lab, and I talk about it in our reviews like I'm doing right now. But this would fall into the firmicutes, which are gram-positives, not the gracilicutes, which are gram-negatives, not the Mendoza-cutes, which are archaea, or our Tenericutes, which are our mycoplasmas. that is it for the review today. What kind of questions do you guys have for me before I have to go? I've got 15 minutes before I need to leave. Any questions? All right, you guys are studying. You know the drill. So as you're studying, if you come up on anything that has you asking questions, email me. And you know that I will email you back as soon as I can. As you're watching the lectures, as I'm sure you're going to go back and do, and you're learning more about this stuff, email me if you have questions.
I have a question.
Yes,
So sorry I was late into it. I couldn't get my audio to work right.
you're fine.
Did you mention anything about this particular training being recorded or not?
Yes, so I will always record my reviews. My Thursday reviews, I will always record them and post the recordings in the discussion board on Canvas.
Okay, because I want to catch the beginning.
Yeah, even if nobody showed up, I will post a recording from a previous semester. So you'll always have something to cover that week's learnings.
Perfect. Thank you.
Yeah, no worries. Any other questions?
Nope. Very informative. Appreciate it.
Yeah, no problem. So like I said, study up, email me, take a nice break, take a breather, and I will see you guys next week. Email me with questions. Thank you guys for showing up.
Thanks.
Thank you.